Good evening. So in the last class we discussed regarding the cell organelles, and now some more cell organelles we will discuss today. The next one is very important cell organelle, that is ribosomes. The cell organelles without membrane concerned, so they are without membrane. This is very important point here you have to remember. They do not have any membrane and they are concerned with protein synthesis. These are called ribosomes. They were first observed by George Palade in 1955. So sometimes they are called also Palade bodies, but now they are ribosomes. They occur in both prokaryotic and eukaryotic cells. So both the cells we can find the ribosomes. In prokaryotic cells, the ribosomes often occur freely in the cytoplasm, whereas in eukaryotic cells, they remain free or attached to the outer surface of the endoplasmic reticulum. Hence, they are called a rough endoplasmic reticulum. Since they are attached with the endoplasmic reticulum, they are called a rough endoplasmic reticulum. A large number of ribosomes occur in a cell. For example, a single cell of bacterium E. coli contains nearly 20,000 to 30,000 ribosomes in a single cell. Their number in eukaryotic cells is several times more. Further, the cells which are involved in active protein synthesis contain large number of ribosomes. Generally, the cells where the protein synthesis takes place having more number of ribosomes. Depending upon the place of their occurrence, ribosomes are of two types. One is cytoplasmic and another is organelle. The organelle ribosomes are found in plastids, it's called plastidoribosomes, and in mitochondria, mitoribosomes. The cytoplasmic ribosomes, these are cytoribosomes, may remain free in the cytoplasm matrix or attached to the surface of the endoplasmic reticulum with the help of a special ribofourin. With the help of this one, these ribosomes are attached with the endoplasmic reticulum. Different types of ribosomes may be produced in different types of proteins. For example, structural proteins from free cytoplasmic ribosomes and globular proteins from ribosomes bounded with the endoplasmic reticulum. So this way there are found two types, structural proteins and globular proteins. Types of ribosomes depend upon their size. The size of the ribosomes is determined by the sedimentation coefficient in the centrifuge. It measured in Swedberg units. It is measured in Swedberg units, generally called the S. Recently, according to size and sedimentation coefficient, two types of ribosomes have been recognized, depend upon the size and sedimentation coefficient. These types are 70S ribosomes. The 70S ribosomes are comparatively smaller in size. They have sedimentation coefficient of 70S and the molecular weight 2.7 into 10 to the power 6 Dalton. They have two subunits. Those subunits used to join, those are 50S and 30S. Regarding these 70S ribosomes, they are found in the cytoplasmic ribosomes of prokaryotes, blue-green algae, and bacteria and PPLO, pleuropneumonio-like organisms. Regarding the second one, that is 80S ribosomes, the 80S ribosomes have the sedimentation coefficient of 80S and molecular weight is 4 into 10 to the power 6 Dalton. There also you can find two subunits of ATS or 60S and 40S. So generally if you see 60 plus 40 it is 100, but it is not the case here. When they attach together, actually this is the area you can take 80, but here you can find 60 plus 40, they combine, they can get only 80S. So the cytoplasmic ribosomes of eukaryotic cells are having 80S type of ribosomes. So here the diagram is given regarding the prokaryotes, the 30S and 50S, there you can find 70S. And in eukaryotes 60S and 40S, there you can find 80S type. The subunits, one smaller unit, another bigger unit, they unite together, they become 80S and 70S. In eukaryotic, ribosomes are synthesized in the nucleus and enter the cytoplasm through the nuclear pores. Each ribosome consists of two unequal subunits. I told you already, the smaller one is ovoid and fits over the larger one, the dome-shaped one, like a cap. These subunits occur separately in the cytoplasm. These subunits are separated. The small one and bigger one are separately found in the cytoplasm and join from the ribosomes only at the time of protein synthesis. At the time of protein synthesis, these two subunits join with the help of magnesium ions. Magnesium ions they used to join. At the time of protein synthesis, many ribosomes line up and join with mRNA chain to synthesize many copies of particular polypeptides. Such a string of ribosome is called a polysome. So look here. Regarding this protein synthesis, you know that you can find a nucleus, and from this message comes through mRNA. This is called mRNA. In the cytoplasm, you can find sRNA is there. sRNA is found in the cytoplasm, or soluble RNA or tRNA. They used to collect. The nucleus fixes particular proteins, preparation of particular protein. So this sRNA collects this material from the cytoplasm and take to the place where you can find a ribosomal RNA or RNA is there. There only you can find chain of mRNA attached together and form the preparation of particular proteins. So proteins are prepared by the effect of this nucleus gives the information, the mRNA get the information and transcribe that information and get into the mRNA, this rRNA, and prepare the food made these proteins. So at the time of protein synthesis, many ribosomes line up and join with mRNA is a chain that is called polysome, is a chain, and synthesize many copies of particular peptides, polypeptides. Peptide is nothing protein, polypeptides are proteins. Such a string of ribosome is called polysome. So here you will get a number of ribosomes are attached and the mRNA is called this polypeptide chain, and then this is a single ribosome. Here you can have a smaller unit and a bigger unit, in between you can find mRNA. Regarding the functions of ribosomes, first one is ribosomes are the sites of protein synthesis in the cell, hence they are popularly known as the protein factories of the cell. Generally they are called the protein factories of the cell. And ribosomes also furnish enzymes, peptides, transferase and factory needed for the formation of the polypeptides. Polypeptides are proteins. So mainly the ribosomes are concerned with the production of proteins. Ribosomes also provide site for attachment of tRNA and mRNA which participate in protein synthesis. Newly formed polypeptides is protected from degradation by cytoplasmic enzymes. Newly formed polypeptide is protected from degradation by cytoplasmic enzymes. Regarding the next cell organelle is microbodies. These are single membrane bound and made up of phospholipid layer, minute spherical vesicles, and range from 0.5 to 1.5 microns, present in plants and animals containing various enzymes. They are first observed by Rodin in 1954 in mouse kidney cells. So these are micro bodies are first observed by Rodin in 1954. It contains a matrix of intracellular materials including enzymes and other proteins. Microbodies are of many types, but we will discuss only two. Those are peroxisomes and glyoxysomes. Peroxisomes and glyoxysomes. So the first we will take peroxisome. The term peroxisome was coined by D. Dew in 1969. They are microbodies which contain enzymes for peroxidase biosynthesis. They are found in both plant and animal cells, and generally they are close association with the endoplasmic reticulum. These peroxisomes are closely associated with the endoplasmic reticulum. They contain special protein called peroxins, and for obtaining the material for cystole and endoplasmic reticulum. Peroxisomes contain oxidative enzymes like urate oxidase, D-amino acid oxidase, hydroxy acid, and B-hydroxy acid oxidases, catalases, etc. they can found. So regarding this anatomy of a peroxisome, here you can find it is a globe-like structure having the wall is limited with the plasma membrane structure and a bilipid layer, and inside you can have urate oxidase crystalline core having many types of oxidases inside this peroxisome. Regarding the functions, they are associated with the chloroplasts. Peroxisomes perform photorespiration. This you will learn the next year. A respiration occurs in the daytime in the basophil cells, that is called photorespiration, and protects the cell from the toxic effect of H2O2, where these oxidases form this H2O2 into oxygen and water. So this toxic effect is reduced by the microbodies like peroxisomes. The next one is glyoxysome. These are also single membrane bound and tiny organelles found in the cytoplasm. Here you note down cytoplasm, CYTO, of certain plant cells. They were discovered by this Braden-Batch in 1967. They consist of an amorphous protein matrix surrounded by a limiting membrane. These glyoxysomes contain enzymes for beta oxidation of fatty acids and glyoxylate pathways. They are quite common in germinating oil seeds like castor, and also in watermelon, cucumber, peanut, and other seeds you can find these glyoxysomes. So remember, generally quite common in germinating oil seeds such as castor and watermelon, other seeds like watermelon, cucumber, and peanut. Beta oxidation of fatty acids produces acetyl CoA. It is metabolized in the glyoxylate cycle to produce carbohydrates. This is another pathway to get carbohydrates through glyoxylate cycle. So here you can find the glyoxysomes. The center, it is this mitochondria, and they have these, all the enzymes are inside these glyoxysomes. These are found in the cells. Regarding this, some other subcellular organelles. There are many subcellular organelles found in the cytoplasm, as I told. The important one, another very important one, is the microtubules. Microtubules are very good, and in plant cells they were first observed by Ledbetter and Porter. Ledbetter and Porter in 1963. These are unbranched, hollow tubular structures made up of a protein that is known as a tubulin. This may be alpha and beta, two types of tubulin you can find. They are made up of this chemical substance. They are generally about 25 millimicrons width, and the core is of 15 millimicrons and wall of 5 millimicrons thickness. Microtubules is a very important function here. We have formation of spindle fibers, cilia and flagella. The thing is, during cell division, you know, the spindle formation takes place. In animal cells, centriole and centrosome are there. In plants, from which part we can get the spindle fibers, that is microtubules. In plant cells, the spindle fibers are developing from the microtubules and they help in the cell division. Microtubules along with microfilaments constitute a cytoskeletal fabric in the cytoplasmic matrix. So these microtubules also form as a network in the cytoplasm. So that is about these microtubules. The main important of the microtubule is the formation of spindle fibers and which help in the cell division. The cytoskeletal matrix gives rigidity to the cytosol, that is cytoplasm. The microfilaments are made up of proteins like actin and myosin and tropomyosin and others. So here you can find the microtubules, the chains or bundles, they are single here, microtubules, and made up of this, the red color here, that's called alpha tubulin, and green, this yellow color beta tubulin. It is made up of like this. In a cross section we will get the microtubule like this. These are the microtubules here also given, and having around a spiral way this tubulin is arranged. Here also the alpha and beta tubulins. This is the transverse section of a microtubule. Okay. So far we discussed different types of cell organelles, their structure and their function. Now we will go to the last phase of this cytobiology. This cytology is the cell division. In the cell division, regarding this growth and development of every living organism depend upon the growth and multiplication of its cells. So all the living organisms are developing from, especially the higher plants develop from a single cell, a diploid single cell. It undergoes cell divisions and then cell elongation and then formation of the tissues and then organ and then organism. After developing the organism, we can find number of cell divisions takes place, especially the cell divisions two types, mitosis and meiosis mainly. The mitosis, it increases the number of cells, similar cells having the same constitution and same number of chromosomes. Whereas the second division, meiosis, that takes place in the reproductive cells and a reduction of the chromosomes takes place. We will discuss one by one. So the growth and development of every living organism depends upon the growth and multiplication of its cells. Organisms may be unicellular or multicellular. All multicellular organisms started their life from a single primordial cell called zygote that I told you now regarding the zygote. From in this zygote only cell divisions takes place. So the body of the cell increases by the cell divisions only. Cell enlargement and cell differentiation results in the body organization. So that just I told you these things. So in a multicellular organisms, they started their life with a zygote, a diploid zygote. It is having two sets of chromosomes. From this it undergoes cell division, cell differentiation, and organs and organization is developed. In unisexual organisms, cell divisions help in the asexual reproduction, whereas genetic equality and continuity are maintained by a type of cell division called mitosis. This is another important thing regarding the mitosis. After mitosis, one cell gives two cells, and the two cells having the same genetic constitution of the parent cell. That is very, very important here. In asexually reproducing organisms, the number of chromosomes is doubled per each generation, since it involves in the union of gametes. So, sexual reproduction, where you can find the fusion of a male gamete with a female gamete. Male gamete is having haploid number of chromosomes and female having haploid number of chromosomes. After fertilization, a zygote develops having the two characters, and also the total number becomes double. So that is diploid number of chromosomes are found. This is the zygote. So in sexually reproducing organisms, the number of chromosomes is doubled per each generation, since it involves the union of gametes. Two gametes unite together, so the chromosome number doubles. The chromosome number is specific for each species. In each species, the number is fixed. To maintain the constant number of chromosomes, a special type of division called meiosis. So here, in the what's called two gametes joined together, two gametes here you can have haploid number, another female gamete having haploid number, and the zygote to have a diploid number. Diploid number of zygote is there. So then, for the formation of gametes, again, so is diploid, a zygote fuses with another diploid, will get a tetraploid. That's not the case. Again, in the formation of gametes, it undergoes meiosis and the chromosome number becomes halves. That is due to the meiosis only. That is the significance of the meiosis. So in mitosis, a cell gives a number of cells but having the same chromosomal structure. But whereas in meiosis, it gives the chromosome number becomes half. So mitosis and meiosis are indirect cell divisions involving the formation of a spindle apparatus or spindle fibers. Amitosis is another, that is a direct cell division, where without formation of the chromosomes, the nuclear material itself separates. That's called nucleoid is separate, and it involves no spindle formation. Generally you can find in prokaryotes, especially in bacteria. Cell division involves the division of the nucleus and division of cytoplasm. So in the cell division, the first, the nuclear cell division, where the nucleus becomes 2 or 4, whatever it may be, and then in between the daughter nuclei, separation of cell walls takes place. That is called cytoplasm. This is a division of nucleus, karyokinesis. Division of cytoplasm, this is cytokinesis. If karyokinesis is not followed by the cytokinesis, what happens? One cell becomes 3, two daughter cells are formed, and then no cytokinesis, again cell division, 2 become 4, 4 become 8, etc. You can find a multinucleate condition comes. When there is no cytokinesis, if the karyokinesis is not followed by the cytokinesis, it results in the formation of multinucleate condition. So in the same cell, you have many nuclei. That is a multinucleate condition. Such conditions you can find in the embryos, etc. And endosperm formation, you can find multinucleate conditions. Such a division is called free nuclear cell division. Free nuclear division. So cell divisions generally, you know, a mitosis, and meiosis, but this is also a type of division where the karyokinesis is there, cytokinesis is absent. That is called free nuclear cell division. So the cell division is of 3 types. Generally the free nuclear cell division is not considered, but mainly amitosis, mitosis and meiosis. So in mitosis and meiosis also you can find the formation of the chromosomes, whereas amitosis, no formation of chromosomes. So mitos means thread-like structures, so that is chromosomes are not formed, so it is amitosis. A mitosis is a type of cell division which involves no spindle formation. In this case, the nucleus, generally called nucleoid because there is no nuclear membrane, divides directly into two. Therefore, a mitosis is described as direct division or direct cell division. The division of the nucleus is followed by cytoplasmic division. A mitosis is common in bacteria and other organisms like yeast, amoeba, and nutritive tissues. In bacteria, there is no true nucleus. They contain only one naked circular DNA molecule which divides directly by the replication. In others, the nucleus elongates and becomes dumbbell shaped. And a constriction occurs in the center that we will discuss in the bacteria. A constriction upper deeply in the nuclear side and divided into two daughter nuclei. The nuclei may be not equal. See, the materials may be not equal because this nuclear material becomes two halves, but these two parts may not be equal. They may be unequal size of genetic material found in different cells. So here the diagrams are given. Here you can in the first cell having the plasma membrane and DNA nuclear material, and a constriction occur at the walls. This is the constriction occur on the both sides. The nuclear material becomes off and a formation of cell wall. Two cells are formed. That's all. No formation of spindle fibers. The next, the mitosis. This is discovered by German biologist Walter Fleming in 1882 in animal cells. But in the same year, in plant cells also discovered. But Fleming was regarded as the father of mitosis and cytogenetics. Walter Fleming, he only coined the name mitosis. Strasburger in the same year 1882 discovered mitosis in plant cells. Mitosis is also known as equational division or somatic cell division and vegetative cell division because it occurs in vegetative cells. Mitosis occurs in vegetative cells, so it is called vegetative cell division or somatic cell division. It is defined as the sequence of events. So you have to see the cell cycle, how the cyclic nature takes place and formation of new cells. It is defined as the sequence of events by which a cell synthesizes the other constituents of the cell, duplicates, giving new cells and its genome, and eventually divides into two daughter cells. So starting from initiative stage to the formation of daughter cells, that is called the cell cycle. So he given the cell cycle. Actually these things I will explain later. A eukaryotic cell and having different phases, G1 stage, and then cells that cease, they divide, some cells are dead and remain, you can find the synthetic of DNA, and then formation of the remaining cells and mitosis takes place. In this mitosis you have different phases. So cell cycle is divided into two major phases. They are interphase and mitotic phase or mitosis phase. In interphase is nothing but a preparatory stage for the next cell division. It is much longer and concerned with the preparation of cell for the division. It takes place 90 to 95% of the total time of the cell cycle. Out of 24 hours duration taken by a cell for one cell cycle, cell division takes place only 1 hour, and the interphase lasts 22 to 23 hours. So it is a long duration. It is further divided into three subphases: G1, S, and G2. In addition to normal non-dividing stage, after attaining a particular stage, there is no division. That is called GO. GO phase. So now again I will see the diagram. The first stage you can find the G1, the preparatory phase, and DNA synthetic is called the S phase. Yes, there is DNA synthesis. And G2, the preparation for the mitosis and the division of remaining cell organelles takes place, and then actual mitosis starts. G1, it refers to the first phase of the cell cycle occurring prior to DNA replication phase. That is, DNA replication phase is the S phase. Cells is metabolically active after cell division. They synthesize new proteins and RNA but not DNA, because DNA synthesis takes place separately in this G1 stage, needed for several metabolic activities, and grows in size. When a cell does not enter into further divisions, non-dividing phase refers to as a G0 or GO phase. That is the quiescent stage, inactive but metabolically active stage. The S, this is the main phase, that is the synthetic phase. It makes the period during which DNA synthesis or replication takes place, because in a mitosis one cell becomes two cells and having the equal amount of nuclear material, so it has to double. That structure will take place in S phase, that is the replication of DNA. One DNA becomes two takes place only in synthetic phase. Amount of DNA in chromosomes per cell doubles, but not the number, but the amount increases. So each chromosome has now new chromatids. New chromatids are formed in this stage, and there is a central centromere, and on both sides you will find the chromatids are found in this phase. The G2 phase, during this phase, proteins responsible for the formation of spindle fibers are synthesized while the cell growth continues, and the remaining floral parts also, remaining these cell organelles develop in this phase. So now you see the same diagram you can find again and again. There you can find this mitotic phase, and G1 growth and normal metabolic role, and S phase DNA replication phase, and the G2 growth and preparation for mitosis. Now the mitotic phase, the most dramatic period of cell cycle, but a short phase involving a major reconsolidation of virtually all components of the cell. It is also called equational division, as the number of chromosomes is equal in both parent and daughter cells. So similar amount of cells, amount of chromosomes found from daughter cells. The mitotic phase, though it is a continuous one, it is divided into 4 stages arbitrarily to our convenience. They are prophase, metaphase, anaphase, and telophase. So prophase, metaphase, anaphase, and telophase. So 4 phases. The first one is the prophase. This is the first phase of the mitotic phase, nuclear division following, and interphase of the cell cycle. Interphase of the cell cycle. Chromosome material is entangled during the condensation of the chromatin reticulum. So in this one, the first you have this nuclear material. Here you can have the nuclear material having chromatin reticulum. They become shorter and thicker, and formation of the chromosomes. Prophase is characterized by compact chromosomes due to condensation of chromatin. 2 chromatids attached at the centromere of each chromosome. As I told you already, here you can find there is a central centromere. On both sides of the centromere, chromatids are formed. Chromatids with centromere. Initiation of mitotic spindles. Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope disappear. So in the cell, the chromatin reticulum becomes shorter and thicker, and removal of nuclear membrane, and remaining cell organelles, Golgi apparatus, and endoplasmic reticulum, etc., they disappear. So now you see, in the first phase, it is in allium SIPA actual diagram from a cell. There you can find first nucleus and slowly disintegration of the nuclear membrane, and the chromatin reticulum becomes shorter and thicker, and here you can find the final prophase stage. And also different phases are given. I will explain later. This is the first phase you can have. This is the interphase, the preparatory phase, and next slowly disappearance of the nuclear membrane and formation of the chromosomes. In metaphase, the chromosomes are arranged on an equatorial plate, and then anaphase, the moving of the daughter chromatids to different poles and formation of cell wall, and one cell becomes two cells. So it is obtained from the allium root tip. This is a natural diagrams you can have. This prophase, here you can find disintegration, and later prophase, formation of chromosomes, in the arrangement of chromosomes in the center, in the metaphase, and in the early anaphase just they are moving, and anaphase, final anaphase, they are completely moved to different poles. In the telophase, there you can find the formation of cell plate in the middle, and here enlargement of the middle and formation of daughter nuclei, and two cells are formed at the later telophase. This is the actual diagrams in the actual photography in allium root tip. So regarding the second one metaphase, that's the second phase of mitotic phase, the disintegration of the nuclear envelope and spreading of the chromosomes in the cytoplasm, forming metaphase plate in equatorial plane. Chromosomes with the two sister chromatids attached to the spindle fiber by the kinetochore, that is the center of the centriole. So the center of this chromosome, centromere, inside you have the kinetochores. This phase is the best stage to study the structure of chromosomes. See, this metaphase, this you will get a question in the metaphase. What is the phase where you can see the clear structure of the chromosomes, and also we can count the number of chromosomes only in this phase, that is metaphase. So here the metaphase, here you can find that you have seen already, they are arranged in the equatorial plate. Chromosomes arranged in the equatorial plate. Regarding the next anaphase, to go back, ana means back, to go back, chromosomes going to the back. Division occur in the centromere, and each chromosome is split into two daughter chromatids, and now they are called chromosomes, which begin their migration towards the opposite poles by contraction of spindle fibers. By the contraction of the spindle fibers, the daughter chromatids move to different poles. So each chromosome moves towards the pole with its arms trailing behind, assuming a V shape and L shape. If it is there going, the chromosomes may be move like this, a V shape or U shape, or sometimes you have L shape, they are moving to different poles. Here this diagram, they has completely moved to different poles. And in the last telophase stage, that is the fourth phase of the mitotic phase, chromosomes reach the poles and decondense. Actually they are nuclear envelope and nucleolus reappear. What are the changes that you take in prophase? That is a reverse changes we can get here. See, there you can find this chromosomes become chromatin reticulum and formation of nuclear membrane and formation of remaining cell organelles, and then one cell becomes two cells. So clustering of chromosomes at opposite poles are changed into chromatin reticulum, reassembling of nuclear envelope around the chromatin and nucleolus, and reappearance of Golgi complex, endoplasmic reticulum, and other subcellular organelles. So reappearance of the Golgi bodies, endoplasmic reticulum, and other subcellular organelles. And so far we finished the karyokinesis, and now we will see the cytokinesis. It is followed nuclear division by the formation of a cell wall or cell membrane between the new nuclei, between the two newly formed nuclei. In plant cells, formation of cell wall begins as a phragmoplast. So, in between the two cells, here you can find two daughter nuclei. At the beginning in the center, phragmoplast develops, and this phragmoplast forms at the center and moves towards periphery and formation of two cells. So that is centrifugal wall formation you can find here. It is formed by the aggregation of the vesicles produced by Golgi complex. So Golgi complex has a role in the formation of phragmoplast or cell plate in plant cells. Cell wall material in these vesicles fuse one another to form cell wall, and then pectin may be deposited on the cell walls, and from middle lamella you can find the formation of primary wall and secondary wall, etc. Cell plate beginning from the center and extend to the periphery. Whereas in animal cells, cell plate is formed or appeared in furrows in the periphery, this plasma membrane, and these furrows deepen and join in the center. So in animal cells, if you happen to see the plasma membrane, a furrow appear in the plasma membrane from periphery, and it will enter to the center. So formation periphery to the center, that is centripetal wall formation you can find in animal cells. So again, all the phases are given. The stages of cell division you can find in the prophase, and there you can find the metaphase. All the chromosomes are arranged in the middle, and then moving of the chromatids takes place here, and complete moving here, and formation of cell plate and development of new cells. So now let us see the significance of mitosis. So production of two daughter cells which are quantitatively and qualitatively and identical to each other. Not there? Qualitatively, quantitatively identical to each other and identical to the parent cell. That is very important here. These two cells are identical and also identical with the parent cells. And constants of the chromosome number in all cases of an organism. The same number of chromosomes follow in all cells of an organism. Help in growth and development of multinuclear organisms. They help in the growth and development and asexual reproduction in lower plants. This mitosis is help in asexual reproduction in lower plants. Vegetative reproduction in higher plants by grafting and tissue culture, etc. This is used. Regeneration of lost or damaged tissue and in wound healing also help in this mitosis. So mitosis is helpful in the wound healing and damaged tissues, etc. It is helpful. So this is about the mitosis, and when anything you have to remember here is the cell gives two cells, and those two cells are equally or qualitatively and quantitatively similar to the parent cell. Okay, we'll continue later.